photosynthesis, very similar to plants. Two photosystems. As you can see with the black arrows, the electron flow is linear, starting with water as the ultimate donor. And then it zips through all the way to NADP plus. So, of course, when you have an electron transport chain, you think about proton gradients. They go hand in hand, and that's what you see. The proton gradient getting established across the membrane as the electron flow is happening. And we end up with NADPH and ATP. Okay, then we learned that there's some microbes that use just one photosystem, not two. So here is an example where you still have a linear flow. If you follow the blue arrow, you can see the linear flow of electrons starts from something like hydrogen sulfide. Where did it go? Come on. Here's your hydrogen sulfide. And then you follow the blue arrows and you end up at ferredoxin. And then you get NADP plus reduced to NADPH. And the artist did not put it on here, but this also does generate a proton gradient. Okay, so there is a proton gradient that gets established. So those microbes that can use photosystem 1 only, they get NADPH still, and they get ATP still. It's just a little simpler system. Then we talked about photosystem 2. So there's some microbes that do that. And they give them the cute name purple non-sulfur bacteria. So that's the example I'll give you. They're in the group alpha proteobacteria, which is a huge group that includes things like E. coli and others. So they do something weird. So if they just have photosystem 2, these type of bacteria, what they do is an electron in the bacterial chlorophyll pigment that is part of the reaction center gets excited and ejected off into an acceptor, but then it goes through this circular pathway of flow. And so they call it cyclic photophosphorylation. Cyclic, because the electrons are going in a circle. They start at the bacterial chlorophyll and end there. So it's just zipping around and around and around. Photophosphorylation just means light energy is being used to take a phosphate and stick it onto ADP, get you your ATP. So the other kind of weird thing is that if they do use just photosystem 2, they don't have a way energetically to make any NADH or NADPH. It doesn't work. So they're at a disadvantage. They can get their ATP, but directly they can't get NADH or NADPH. So let's look at a picture. This is if they just use photosystem 2. So this big yellow blob is your reaction center. Here's the bacterial chlorophyll electron getting excited by light energy. And you can see if you follow the blue arrows again, it's just going around and around and around. And so it comes back and then gets hit with light energy and then goes around and around and around and just keeps zooming around like that. And if you look at how they've drawn it, this time anyways, they at least give you a proton gradient idea. And so you do get ATP, so that's good. But nowhere on this drawing do you see anything about NADH or NADPH getting made. And the artist is correct not to include that. So they're a little out of luck. So here, the only thing you see that alludes to it is right here. Reverse electron flow makes NADH. So reverse electron flow, this is something that takes energy. So what they're implying here, let me go to this one. So this is an example of a purple bacteria doing its thing. So this P870 down at the bottom, that's your bacteria chlorophyll, electron getting excited. And they're trying to show that when it gets excited, it boosts up into an excited state. And so now it's up into the negative scale of the reducing potential, which means it's very happy to give off an electron when that chlorophyll is in an excited state. Then what happens is, in order to make any DPH or any DH, you have to be right around here on the reducing potential scale. But it doesn't work out that way. In a purple bacterium, the quinone, you don't need to know the name of this, but the quinone gets the electron and they don't have enough oomph. They're not above here, they're down here on this power. And so they have to do reverse electron flow. So that dashed line is summarizing, yep, they got to do reverse electron flow in order to get NADH or NADP plus or NAD plus reduced. So if they say reverse electron flow and they're showing this arrow going up this reducing potential tower, what that means is this bacteria has to actually use ATP to get any NADH. So they have to put in some energy. So this is kind of a downer. Okay, it's not terribly efficient. So they can make ATP, but then they have to use a little bit of ATP to get any NADH if they need that, which they will. It's still considered anoxygenic because nowhere on here do you see oxygen being made. When any electrons are getting stripped from anything, there's no oxygen generated, so it's still without oxygen. So these bacteria tend to live, oh, in here too. Because they need to really be as efficient as possible, they tend to have an interesting cell structure where inside their cell, they have this membrane material that they call a thylakoid, which is what we call the inner membrane inside a chloroplast. And so it's similar idea, although we still don't call that an organelle inside that purple bacteria, but it's a nice infolding of the membrane so that they can stick all of the bacteria chlorophyll and all the complexes. They can really jam it full so that they can do as much as they can with ATP formation. So they try to make up for it in terms of just beefing up their cellular membrane so that they can really pack it jammed full and get a lot more because of that increased surface area. So here we're looking at 3 different groups of bacteria, and these bacteria are basically trying to create a way to get ATP made and hopefully some NADH or NADPH. I'm not going to go into details on this. Here they're showing the purple bacteria doing, oh, they got to do reverse electron flow if they want the NADH. But basically, you can see that they're on the reducing potential tower again, or redox node, and you can see that purple bacteria are a little bit lower on it, so they're not quite as efficient. Whereas green sulfur and heliobacteria, they're a little bit higher up. So it's a little easier for them to work through this photosynthesis and get more bang for their buck. So this is all fine. This is the light reactions of photosynthesis going on. And the other part of photosynthesis is the second phase, I call it, which is the Calvin cycle or the C3 cycle. All right, so the Calvin cycle or the C3 cycle, that's part 2 of photosynthesis. That's how they make their sugars or carbohydrates. And depending on the microbe, they may have to use ferredoxin in order to get their sugars made. So that's one reason why I'm showing that. So I'm not going to ask you to memorize this in any way other than to know that the Calvin cycle is part 2 of photosynthesis. You got to use some ATP from the light reactions for key steps of it. and NADH or NADPH as well. And so with the Calvin cycle, what you see here is CO2 being brought in, and they run this cycle, and here where it says to biosynthesis, what they mean is this is where they create their sugars. Okay, so they use this cycle, and then once they get to enough of this glyceraldehyde 3-phosphate, once they get enough of that, they can send it into a separate pathway to make sugars. And then they got to keep the cycle going all day long so that the CO2 that gets brought in can keep getting fixed into sugars. So most bacteria will do this, just like plants do this. So cyanobacteria do this, most bacteria do this, but not all. There's this weird, oh, this is another example of the Calvin cycle. So I just either or, it's the same thing, just whatever one you like to look at. Some microbes do this funky thing where they use ferredoxin, and they have to do that in order to make their sugar. And they take the citric acid cycle and put it into reverse. Really weird. And so here they actually call it the reverse citric acid cycle. So that's what you're staring at here. And so this is the citric acid cycle. The microbe just says, I want to reverse it. And so here you can see, instead of the citric acid cycle producing CO2, like what we talked about, here CO2 is getting pushed into it in reverse with the help of ferredoxin to help with this process. And then we end up with your sugars. So the hexose P and the cell material, those are your sugars that ultimately get made. So there's some bacteria that do this. You don't have to know who does what. I'm not asking you to memorize which bacteria do what, but just understand that these funky bacteria, some don't run the Calvin cycle. They take the citric acid cycle and reverse it. So pretty interesting. Okay. Well, as far as photosynthesis, that's basically where I'm going to stop. We're just trying to get across the point that there's more than one way to do photosynthesis when you get into the microbial realm, and that you have options. It's not always the Calvin cycle to get the sugars. Sometimes they don't use two photosystems when they're doing light reactions. So as you're studying this material for the third exam, please do, I think the best way to handle this is to do a compare and contrast. Look at how, these different groups are doing it. What's similar, what's different, what's the payoff of it? Is there any downside as you're studying this? So I think that would help, because it'll get you to see some of the details here on the important goals and information. So to finish up this slide set, there's some terms that I wanted you to be comfortable with, not for this exam on Friday, but the next one. So when you look at microbes, whenever you are trying to get to know a microbe and figure it out, you want to know how does it get its carbon? Since microbes are carbon-based, they need carbon. So how are they doing that? Where are they getting their energy from? And where are they getting their electrons from? And so I've grouped these terms into pairs. So microbes, they can either be autotrophs or heterotrophs for their carbon source, right? Autotroph is like what we just talked about. If they can do photosynthesis, for example, or if they're a lithotroph and can take in some CO2, then they're considered autotrophs. Heterotrophs, that's us, we're heterotrophs. We have to eat something that is carbon-based as our source of carbon. Then the next two pairs are here, phototroph or chemotroph. That's your options for energy. Anything on the face of the earth, they either use light for energy or chemicals. We have to use chemicals. Unfortunately, we can't just lay out on the beach and get our needs met that way. And then the other two pairs are lithotroph or organotroph. So that's the electron idea. What's their source of electrons? It's either going to be inorganic or organic. Those are your options here on Earth. And so lithotroph is an inorganic electron source. And we're organotrophs. We have to use some kind of organic material as a source of electrons. So again, memorizing these in pairs and understanding, how are they getting their carbon? How are they getting their energy? How are they getting their electrons? This is a huge concept in terms of, this covers it all. All microbes fit into these categories somehow. And so now what you can do is start taking these terms and smushing them together. And that's what the next slides are. So if you, for example, talk about a photolithoautotroph, if you pick that term apart, well, okay, photo, that must mean it's light energy. So that's their energy source. Litho, that's inorganic. Litho literally is referring to rock. So inorganic electron source. And then if they're an autotroph, they can make their own sugars. So they're taking in CO2. So cyanobacteria fit this. Purple and green bacteria fit this. Then you can say, what if you smush these terms together? Chemo, organo, heterotroph. So now chemo means it's chemicals that provide the energy, and organic for their electrons and carbon. So, whoops, we fit that. We fit that category, so do a lot of pathogens, of course, because pathogens like to hang out with similar based organisms. So that's why they can hang out with us and cause problems for us. Fungi fit into here, many protozoa. Okay, what else? Then, and then most organisms that have been studied, the bacteria especially, they fit into one of these two categories. So these are the most common ideas out there in terms of how microbes are living their life. But who knows, that could change over time once we get to know more of the microbes in more detail. Photo-organo-heterotrophs. So these are weird. They use light energy, but instead of taking in CO2, most of them grab an organic source nearby and take that in. Some can do CO2, but most seem to just say, I'll just take an organic chemical and bring that in and use it for their energy. Their electron and carbon source. So these you tend to find in really polluted areas, especially lakes and streams. You'll see quite a bit of these organisms because they can still access sunlight, but they're surrounded by pollution, organic, herbicides and pesticides and other, runoff from soils and so forth. And so they're happy in those environments. And then chemolithoauto, chemicals for their energy, inorganic, inorganic for their electron and energy. And then auto means they can take in CO2. A lot of chemolithoautotrophs also love to strip electrons off of iron, nitrogen, sulfur. Those are favorites, but there's others. So these bacteria we have to thank for how the earth exists today in the sense of all the recycling that they do with the different, the carbon cycle, the nitrogen cycle, iron cycle, sulfur cycle, and on and on and on. So these organisms are pivotal for creating the earth the way it is. And the atmosphere, of course. So not for this exam, but give these slides some thought and think about the, starting to smush these terms together and being able to pick apart what does that really mean. Yeah, no, it's jargon. But once you learn the basics, it's fairly easy to pick them apart. And then I put on some slides here just in case you're interested about life on Mars or other planets. They're really looking into this. This is a paper that was published a couple of years ago, and they were proving here on Earth from rocks they collected from Mars that microbes can grow on those rocks that are here. And so this was some evidence, early evidence showing that Mars does have the habitat that could sustain microbial life. And that's what they think is, was the first thing on Mars. If it was anything else, for sure, they think there was some kind of microbe. And they are alluding to the idea that they're finding more and more evidence. And so this is not going to be on the exam. It's just if you're curious about that and like to think about life on other planets, that is interesting. Okay. I think I'm going to stop there with, I'm not going to start microbial growth. We'll do that Monday, I guess. So Friday is the exam. So let's start thinking about that. And it starts back with cell structure when we get into the peptidoglycan and moving forward from there. So I'm going to pull up that part 1, I think. Yeah. Okay, and then so right now, the draft of the exam that I have is sitting at 100 points. And roughly 60 points are the multiple choice and 40 are from the essay short answer, fill in the blank. Let's go to cell structure and function where the peptidoglycan material is. And let me do this. Yeah. All right. So the material starts here. So we already covered this one, but we're on to the cell envelope for gram-positive and gram-negative. So cell envelope, cell wall, same thing. So working through these slides, are there questions about peptidoglycan or any of the other content on? Pardon me? Together. Yeah, so that's part of it. I mean, some of them anyways can anchor all the way through. Some only go part way. And so that does help give some structural integrity, I guess, to that. That's with the gram-positives.
What's their function otherwise?
Good question. I think some of it is they're I wonder if some of it is assessing their environment, like they are using it as part of their assessment. I don't know. I won't ask you that, because I didn't really get into that in the material. Anything on peptidoglycan? Yeah, probably. So what do you think? If I were going to ask a question on content in chapter 3, cell structure and function, what do you think might be something that could be an essay question? What topics? Yep, gram-positive versus gram-negative, like their cell wall structure, certainly. Compare and contrast. I'm kind of a big fan of that. So that could be something. Yes. Yes, so peptidoglycan structure, although I'm not I'm not gonna get into like, I'm not gonna ask you to memorize which amino acids per se, but I want you to recognize them. So, um, like you don't have to come you don't have to come up with this just out of your head, but recognize it. So multiple choice essentially. But, you know, peptidoglycan structure and the basics of that and like how does it work? How does it go together? That's all fair game.
Do we have to run like the whole Coachella murder protein?
That would be a good topic too, right? Because we did cover quite a bit about that. We looked at fair detail. So being able to talk about the flagella would be important as well as the peptidoglycan.
It may not be up to that. That the protein gradient through the mock protein, divided with the T protein is on the
Yeah,
C ring, T ring.
yeah. So you don't have to know the names of the rings, you know, that's not really, I won't do that, but knowing that there are rings that exist. So that's in the next, hold on to that thought. I'll go into the next slide set in a minute here and then we can talk about the flagella a little bit more. Anything with this though, with peptidoglycan, gram positive versus gram negative. Do archaea have peptidoglycan? Nope. No, they do not. They do the NAG and NAT, so that's not true peptidoglycan. Yes. It might be nice that you know beta 1,4, especially because that's what our lysozyme attacks. It's kind of a cool thing to know. So the beta 1,4 linkage is with true peptidoglycan, and that is what our lysozyme can attack and break down. Well, let's get into the second slide set on cell structure and function. That was looking at the flagella. So there's a question about that. So knowing the structure, I won't ask like, where does the C ring go or the L ring? That don't worry about that, but just knowing that there are rings involved. And then this is, yeah, so here with this proton gradient and the Mach protein, that's an important concept because that's how they get their flagella. How does the flagella move, by the way? Yeah, it rotates, right? So the flagella rotates. So it's not like sperm flagella that whips. It doesn't do that, it rotates. Knowing the types of flagella, like the Paratrichus, polar, Lophotrichus, those would be important. Not all microbes do it the same, and I have no idea why why some microbe would decide to do Paratrichus. That just seems like so much effort. But some do, including E. coli. So we also got into other movements besides flagella, so know some options. It's not just flagella that allows them to move from point A to point B. You got the gliding, and we have the pili structures, and sometimes they use proteins embedded in their cell membrane and wall. Sometimes they just use goo. And then the gas vesicles, if you're aquatic, that's a really good option. Questions on any of that? Talked a little bit about biofilm and like the glycocalyx, the capsules and slime layers. And then if they're all gathered together, we start saying, oh, look at that biofilm. We talked a little bit about that, and then endospores. So again, I'm not going to ask you I showed you the life cycle, so to speak, of the endospore, the formation of it, but that's not something I'm focusing on. I put this up more to show you it's a multi-stage process. And once they kick into it, they can't really back out of it. They got to keep going.
With the vaccine, that can be this any type of like sense that we're gonna apply.
Yes. Yeah, you got it.
Are we looking at the structure of the type 4 villi?
No. So type 4 pili is the idea. I put that in there as a way to move, but we didn't really get into details of that. What else? So endospore structure. So this one here. Like think about the structure. So this is the very outside of the endospore. It has exosporium, mostly protein, and then you can have many layers of these so-called spore coats, more protein. And then you have some loose peptidoglycan, and then inside that is the actual like cell that's there and can become active once the endospore can get into an area that it's okay to go ahead and germinate and grow.
Want us to know more about the chemicals of the endospore.
So yeah, they do have some unique chemicals, right? They got the dipicolinic acid, and so those structures like so for example, if with the idea of dipicolinic acid, it doesn't seem to be showing up in a normal cell, but it is in inside an endospore because it seems to help protect and help protect both the like the structural integrity of the DNA. Plus it helps just deal with not having any of the dehydration going on, harm anything. And then the SASPs are kind of the same thing. And so those two chemicals in particular seem to be really useful for the endospore to be able to withstand really tough times. And then when times get better, they're still good to go. They're protected and they'll be able to germinate and grow. Whoops. Anything else? Yes.
Textbooks, specific question for any of the chapters.
This time around. No. No. There's no like particular page range where I haven't talked about it, but I want you to know. So use the textbook as backup. So remember, with all of my classes, no matter what class I'm working with you all, it's the PowerPoints. That information is number one in terms of focusing on that. Besides cell structure, we also got into metabolism, so we better cover some of that too. Metabolism. So this is where we were looking at, first just orienting you to glycolysis and the citric acid cycle or the TCA, and then the electron transport chain with the idea that hopefully you've had at least some taste of that somewhere. And it creates a common base knowledge for you to build off of. So what's still bugging you about glycolysis or anything else in this slide set? What do you need clarification on?
Are you gonna want us to know how many pyruvate are made from stage 2?
No, how many ATP though? We went over that in a Google Forms question, right? What is the net ATP when glycolysis? They make 4 during stage 2, but they have to use 2. So you only net 2, which is rather pathetic, but it works. Um. Any other questions on glycolysis? Yes,
Glycolysis is just an anaerobic process, not respiration.
correct,
Respiration is a general electronic cycle.
correct. Yep. So glycolysis is anaerobic. How is the ATP being generated? What's the process in general? Substrate level phosphorylation. Substrate, did you have a hand up for a question? Pardon? Oh, okay, go ahead.
I was just wondering, do we need to know like all the fermentation reactions and all that? Like,
No.
just for the,
So there's a couple of reasons why some microbes might go, okay, I'm gonna do glycolysis, I'm gonna do phase 1, okay, I'm gonna do phase 2. I'm getting the pyruvate. Why would a microbe then say, okay, I'll do stage 3? What's going on here? They're gonna stop and assess things. What are they looking for? Oxygen. They're hoping for oxygen. If they can use oxygen and not be poisoned by it, that's what they want. But if they're in an anaerobic environment, then they might do this. They might go to stage 3. That's not the end of the world. Couple of things happen. NAD plus gets generated very often. That's good. Because that can feed back here and basically keep this thing going. So if you're a microbe and you don't have oxygen, you better keep this going and going and going. Because you only get 2 ATP per run. So you want to do this a lot. So having this NAD plus, being able to feed back and keep it going, that's a good thing. Plus, the redox balance idea, remember with fermentation, no matter what fermentation pathway you're looking at, the electron numbers have to equal out. Whatever you started with is what you have to end with. In order to play the game and adhere to all the chemical rules, you gotta meet that. And so that redox balance is also performed during this stage 3. And sometimes a little extra ATP if they go really wild and get into some interesting pathways. So glycolysis is substrate level phosphorylation, and it's anaerobic. And then we got into the TCA cycle. So once the microbe does glycolysis and takes glycolysis to pyruvate, and if it says, oh, I have oxygen, so then the next step they'll choose is the TCA cycle. And so that's why you see pyruvate coming in here and it starts. So I do want you to know the start of this, the idea that you have pyruvate getting converted to acetyl-CoA here, and then you have oxaloacetate reacting, and so your first product is citrate. And then other than that, some of the key benefits. What are some key benefits to what's happening here for the microbe?
ATP.
Yep, ATP or GTP is getting made, and that's actually by substrate level phosphorylation, although they don't show it, the artists be able to show it, but it's happening. And then NADH or NADPH, that's good. FADH2, that's good. And then a bunch of CO2 being given off. That's kind of a hidden good. It's not like obvious why CO2. You know, why would we care? But we care because actually that means the microbe is getting good bang for the buck in terms of it started with glucose, it's breaking that all the way down to CO2. So you can get a complete breakdown like that, you will get more ATP out of that process. So then we care about those NADH and the FADH2, and of course the ATP. Because then the microbe will say, okay, now I've got my NADH, I got my FADH2, now I'm going to link that into the electron transport chain system. And this is where I really win. This is where I get the gold star, because if I can run this, I'm going to get a boatload of ATP. Okay. So again, you don't have to know the agonizing details, but knowing that, how does it start? What do you got with citric acid cycle? Here playing a role with the NADH and the FADH2, those are coming into it. They're donating electrons and protons. And then you get that idea. So here, now bear with me here. I'm going to, let's see if I can pull that up. Bear with me here. So the way I learned this whole idea of the idea of linking the electron transport chains, the electron flow, the proton pumping. So the way I learned it is thinking about, here's another image of that. So I like this one because they're actually showing the ATP base here. And so here you have the electron flow going on with the NADH and FADH2 donating their electrons. So the red arrow is the electron flow. You happen to have oxygen as the final or terminal electron acceptor. And then you can see the pumping of the protons. So remember, this is on a reducing potential scale. So you go from really nicely negative to less negative and then into the positive. So basically what that's saying is you have at the start, these electron carriers are really happy to give off electrons. And then as you get further down here, they're just really greedy for electrons. They're really good at taking electrons. So it creates a really nice situation where the electrons keep moving forward, so to speak, or going downhill to oxygen. And at key steps, there's a little bit of free energy to allow for the pumping of protons. And so I have this in the notes section in previous slides, but I put it here. I think, and this is the way I learned it. I likened it or used the analogy of sledding. When I was a little girl, we had a dog that loved to try to get on the sled as you're going downhill, right? And so the way I memorized this or learned it, I told a story that here's your electron donor or carrier on the sled with the electron, and the dog is the proton. So once you start sledding, you have that energy, you're going downhill, right? You got that energy going. And imagine the dog now catching up to the sledders and jumping on. And now it's got that ability, because of this energy, you're able to get the proton pumped across the membrane. So here's a bunch of protons with the sled dogs. Okay, they made it across, they got pumped across the membrane, and now they're racing to find the ATPase. And once they scoop through the ATPase, then the ATP gets made. So I know this is maybe weird to your brains, or you think it's ridiculous, but I told myself that story years ago, and I still can clearly remember the process. So these types of things, if it helps you to create a story out of it, rather than just memorizing step by step what's going on, storytelling is a very potent way to learn material. So think about that. And then I just love that picture. Isn't that cute? Talk about joy. I mean, both the dog and the kid, just joyful. I don't know who this is, but it was a free picture I had access to. So anyways, so think about those types of ideas as you're looking at the electron transport chain. You are, linking together this idea of, where did it go? Electron flow, providing the mechanism for proton pumping, a little bit of energy. questions on that idea? What about some of the other? Metabolic reactions. We got into the redox tower. How you feeling about that? We did some of the Google Forms questions on this, but. Yes. I don't know. I would give you the information you need, like kind of similar to what I did with the Google Forms question where I gave you. I can't remember, CO2 and acetate, I think maybe I gave you or something, I can't remember. And then I give you the other pair. And so it'd be probably similar to the Google Forms question that we did in class. I probably wouldn't give you the whole tower, you don't really need it. Other questions on? Redox reactions, and we did more fermentation ideas. Redox balance. Oh, and then that funky thing without substrate level phosphorylation. This is really weird. I'd like you to know that this exists, because this is really weird. This is very atypical, but leave it to microbes to whip it out of the hat to do something different. And then the last slide set, remember now, we don't go through the whole last slide set on the part 2. That only goes through the halobacterium slide. But here we got into anaerobic metabolism where oxygen's not the terminal electron acceptor, it's something else. We got into lithotrophy. Anammox, thanogenesis, and then we stop here. Questions on any of that? Okay, well, I'll be again watching my email through the evening hours too. Tomorrow is lab, tomorrow's Thursday, right? Tomorrow, noon lab. I'm your teacher. I'm gonna be there. Isabella has to go out of town, so I will step in and take over. So I will see those of you tomorrow at noon. But send me an email or stop by for office hours if you need to. Or in the evening, if you have a quick question, I'll try to answer it as soon as I can. Okay? And we'll see all of you on Friday.